Hi, I'm Amiko Uchida, and I'd like to tell you about our work on the IL-33 receptor, ST2, and type 2 cytokine expression on tissue eosinophils in eosinophilic esophagitis, or EoE. EoE is a chronic allergic disease of the esophagus characterized by eosinophilic inflammation and barrier dysfunction. This inflammation can also result in fibrosis. The pathophysiology is not well understood. However, several cell types highlighted here are thought to be involved in disease pathogenesis. Importantly, the cytokine IL-33 is elevated in the esophagus of patients with EoE, but the downstream cells and signaling that can respond to IL-33 has previously been unknown. We therefore hypothesized that the receptor for IL-33, ST2, is present on immune cells in the esophagus, and that these cells in turn can respond with downstream type 2 cytokine signaling. To test this, we examined esophageal biopsies from patients with EoE and looked for immune cells shown here. We found esophageal eosinophils specifically have the highest expression of ST2 compared to other immune cells in the tissue, as well as compared to blood eosinophils. We then examined for the presence of downstream effector type 2 cytokines such as IL-4, 5, and IL-13 in these cells. What we found is that eosinophils appear to be major expressors of these cytokines, and we showed this both by flow cytometry at the top of the screen as well as by immunofluorescence, with the cytokines denoted in red and an eosinophil marker denoted in green. To determine if these type 2 cytokines were dependent on eosinophils, we utilized a mouse model of EoE, where exogenous IL-33 is administered and results in esophageal eosinophilia, shown to the right of your screen. We used both wild-type and eosinophil-deficient delta double gamma mice and performed multiplex cytokine analysis on the esophagi of these mice. We found that IL-33 induced IL-4 was critically dependent on the presence of eosinophils, as shown here in the bottom left. Our data support an important role for IL-33 and tissue eosinophils as inflammatory potentiators in EoE. Thank you for your time.